Hello, my dear students. I, Dr. Swati Pandya, welcomes you all in this session. Today we are going to start with chapter 4. It is about animal kingdom. So, animal kingdom is not a new chapter. You must have learned earlier in your primary standards, in your 9th standard. So, here we will discuss about the classification of animals. As we have discussed classification of plants in previous chapter, here we are going to discuss about animals. There are so many animals around us. We know many of them, but we are unknown about others. How they are categorized, what are the different bases, that we will discuss. Before starting classification, let us see some of the criteria which we have to keep in our mind before classifying animals. So, all the members of Animalia, they are actually multicellular, that we have discussed earlier according to Whittaker classification system. All of them do not exhibit the same pattern of organization, but different levels are there. So, what are the levels of organization that we will discuss? What is the type of symmetry? What are the different type of cell, their arrangement, either cellular level, tissue level, organ system, or any other type. Then we have to consider about presence or absence of Silom. Then we will discuss about the type of segmentation. We will discuss the presence or absence of Notochord, like this. So these words are important to learn, like levels of organization, symmetry, presence or Silom, about segmentation, and about Notochord. Let us discuss one by one. First of all, we will see the example of sponges. So, in sponges, the cells are arranged as loose mass of cell and they exhibit cellular level of organization. So, very simple type of organization is seen, very basic concept. Now, some of the division of labor, that means the work is distributed, the function is given to each and every tissues. So, they occur among the next higher group, that is in the coelentrates, the arrangement of cell is more complex, and here the cells can perform their function accordingly, which is given at the level of tissue. So, tissue level of organization is seen. Then if we move to still higher level of organization, that is organ level, and it is exhibited in all the members of platyhelminths, which is higher than these two, sponges and coelentrates. So, it shows organ level of organization. Then if we see next the organ system level of organization, like in mollusk and so on, in all the chordates, even human being, that is included here. So, higher animals, they will show organ system of development or organization. So, in this way we can say that all these animals, according to their basic feature, that is according to the type of cell, their activities, their functions, we can divide them and we can understand their developmental sequence, because on the basis of evolution only we are making the classification system. The first group, that is most primitive, and the last group, which is on the right hand side, that is most advanced. So, here human beings, they are considered in most advanced type of organism. Even different animals, they exhibit different patterns. For example, digestive system in platyhelminths, it has only one opening, single opening to outside of the body, which serves as both, either you call it mouth or anus. So, the digestive system is of incomplete type. This is a peculiar example. But in others, that means Annelida onwards up to chordates, they have complete digestive system, because two openings are found, which is mouth anterior opening and anus posterior opening. Similarly, if we discuss about circulatory system, they are of two type, open and closed. Open type of circulatory system, if we see like in arthropods and some of the mollusks, we come to know that blood that is pumped out of the heart, and directly all the cells and tissues, they are floating inside it. But if we discuss about closed circulatory system, then the blood, it travels into the capillaries, the blood vessels which are known as arteries, veins, and a channelized system that is found. So in this way we can group on the basis of circulatory system also. Now we will understand what do you mean by symmetry. So symmetry in animals, it can be categorized on the basis of symmetry. But what is symmetry? Now by discussing few examples, you will come to know what do you mean by symmetry. First we will discuss about the sponges. Sponges are mostly symmetrical. That means any plane that passes through the center does not divide them into equal halves. Actually, the animal is known as symmetrical when if it is cut through the center, it can divide into two equal halves. But sponges, they are asymmetrical. When any plane passing through central axis of the body divides the organism into two identical halves, then it is known as radial symmetry, like in coelentrates, in ctenophores, and in echinoderms. I will show you afterwards the diagram and you will come to know how we can divide it. But in this way they are divided. Now in animals like Annelida, Arthropoda, and even Chordata, where the body can be divided into identical left and right halves, that means in two sides, and they can be cut only in one plane, then they are known as they have bilateral symmetry. So in this way, on the basis of symmetry, we can define, we can group all the different type of animals. So asymmetric, they cannot be divided into two equal halves. Radial symmetry, when the organism that is divided into two equal halves from more than one plane, and lastly at only one plane, then it is known as bilateral symmetric animal. So here in this page I have explained you how to write all the point wise, main concept based, cellular level, tissue level, organ level, or organ system level, then type of symmetry you are supposed to learn. Even afterwards I have explained you all the things in chart pattern, so you just go through it and try to understand and remember all these things. So now students, we will see in these two diagrams that how the symmetry it can be explained. So first diagram, it is of Hydra, and as the structure of Hydra, that is cylindrical body, so from any plane if we cut the portion, it can be divided into two equal halves. See here, this is one of the plane, so if we cut like this, then two equal halves they are formed. Similarly, this is another plane. So if more than one plane, the animal's body is cut and it can be divided into two equal halves, then we can say that the animal possess radial symmetry. This example is of crab. So another example of crab, it is showing bilateral symmetry, because let us see, with the central plane we have divided the crab into two equal portions. So in crab, when we divide this animal from central one of the axis, only single axis, then only we can divide it into two equal halves. So we have understood about symmetry. Now we will understand about diploblastic and triploblastic type of organization. They are actually based on germinal layers. During the embryonic development, these germinal layers are present. So animals in which these cells are arranged in two embryonic layers, that is outer ectoderm and inner endoderm, only two germinal layers are present, then the animals, they are categorized into diploblastic type of organization, like in Coelentrata. But if there are three germinal layers, outer ectoderm, inner endoderm, and in between the mesoderm is present, then the animals, they are categorized into having triploblastic type of organization. For example, platyhelminths, annelida, arthropoda, echinodermata, mollusk, chordata. So every organism that is considered after platyhelminths into triploblastic type of organization. We will understand this concept according to the diagram afterwards. Let us start with another fact or another basis of classification, that is presence or absence of Silom. Actually, what is Silom? First of all, we have to define it. Silom, it is a cavity between the body wall and the gut wall, and it is very important basis of classification. So, accordingly we will categorize into three subtypes: coelomate animals, pseudocoelomate and acoelomate. Let us see for coelomate. Definitely, if the body cavity is present, the coelom is present, then the animals, they are considered as coelomate, like in annelid, mollusk, arthropod, echinodermata, hemichordata as well as chordata. So all the higher animals, they are considered as coelomate. Let us discuss about another term, pseudocoelomate. Here in some of the animals, the body cavity is not lined by mesoderm. Instead of it, certain scattered pouch are present between ectoderm and endoderm. So actually mesoderm is not conformly present or perfectly present. So such category is known as pseudocoelomate animals, like in escalments. Lastly, acoelomate. Some of the animals, they have totally absence of body cavity. That means body cavity is not at all present. So they are categorized as acoelomate. This concept is even not found. They are most primitive type of animals as compared to the others, like in platyhelminths. So platyhelminths will lack any type of body cavity. Now let us see another criteria, that is segmentation, segments, partitions. In some of the animals, the body is externally as well as internally divided into many compartments or segments with a serial repetition of last some organs. For example, in earthworm, you might have observed also, the body shows this type of pattern. So the pattern is known as metameric segmentation and the phenomena is known as metamerism. So this is a special example, you have to remember it. Another important term is notochord. What do you mean by notochord? Let us define it. Notochord is actually mesodermally derived rod-like structure which is formed on the dorsal side during embryonic development in some of the animals. Animals with notochord, they are definitely known as chordates. We are also one of them. And those animals which do not form this type of structure, they are considered as non-chordates. Like mostly invertebrates, the lower organisms, they are considered in a non-chordate group. Okay students, so now with the help of this diagram which shows the differentiation of germinal layers, and on the basis of germinal layer arrangement we can decide that the animal should be placed in which type of group. So first diagram, it shows diploblastic arrangement. Here this is ectoderm, outer membrane, this is endoderm, inner layer, and how they are arranged. So this is the innermost part which is showing endoderm, then this is the ectoderm portion where the cells are present, and in between the mesoglia is present which is having non-cellular part. So it cannot be considered. So here only two germinal layers they are observed, ectoderm and endoderm. In next diagram, this mesoderm, it implies it takes the position in between these two germinal layers. So here ectoderm is present, endoderm is present, but inner portion which I am showing right now, it comprises of mesoderm. So in total 3 germinal layers, they are showing triploblastic type of arrangement. Now next diagram, it is showing the sectional view of the different arrangement found in different type of animals, and here we are making the groups on the basis of presence of coelom. Silom, you all know, it is a body cavity, and this portion which I am showing right now in diagram A, that is showing the coelom portion. So coelom is present just in between the endoderm as well as mesoderm. The endoderm portion, the mesoderm, and outermost ectoderm. So this is ectoderm, this is mesoderm, and this is the endoderm portion, and here the central cavity that is present. Now if we see the B diagram, a little type arrangement is different. Here I am showing you, I am highlighting these circles. These circles are nothing but these are scattered pouch which are present in the mesodermal portion. So this is actually pseudocoelom, because the coelomic arrangement, it is not perfect, but it has just started. So a false appearance of coelom, it appears in this particular group, and it occur in esculmens. If we see the lower diagram, the coelom is totally absent in C diagram, which shows a coelomate animal. And this cavity which I have shown you right now, that is the gut, which is also known as enteron, and it is for digestion. It is digestive area. So actually body coelom, that is body cavity, is totally absent here. Best example is platyhelminths. So students, here I have explained you how to write point wise, and examples are very important to learn. There are so many terms, and many terms will be confusing you, and I know that this classification of either plant or animal, it is not your favorite chapter, so you will find a difficulty to remember all the things, but write down continuously. Do remember all the examples. This page shows how to write about coelom. The diagrams are important to draw, which I have explained you here. So each and every diagram which I have shown you, that is important, and even you have to revise this topic regularly. So hope you like today's session, and we are ending our session here. Do remember all these terms and take care.